Hi, welcome back to Chapter 10. This chapter is called Preparing for Sexual Reproduction Through Meiosis. And if you remember last week, we talked about mitosis, which is a type of cell division. This week, we'll talk about meiosis, which is a different type of cell division. And in section 10.1, we will go over an overview of this process, and then we will go through the steps of meiosis in more detail after that. Let's do a little bit of review before we get started and go over the terms that we will hear throughout this chapter. So first, let's look at chromosomes and chromatids. And the picture here shows unduplicated chromosomes. So if you remember, there's a phase in the cell cycle where the DNA duplicates. But when you see a single chromosome like this, this is at the start of our process, you have one chromosome from dad and one chromosome from mom. And you have that for each different type of chromosome. Remember, you have 23 types of chromosomes. So there are 23 of these from dad, 23 from mom. So here's one from mom and one from dad. Here's a second one from dad and from mom. And again, you would have 23 of these chromosomes from mom and dad. They call these a pair because this is chromosome 1 from dad and mom. This is chromosome 2 from dad and mom. And again, you'd have 23 of these pairs from dad and mom. Now, when the chromosomes, when the DNA duplicates, we get these pairing up, or we get these chromosomes that have sort of the two sides to them. And they call each of these two sides sister chromatids. So when you see this single line, that's your unduplicated chromosome, but you still have one from dad and one from mom. When you see the two sides here, the two sister chromosomes, you still have one from dad and one from mom for each pair. So for pair 1, you have a chromosome from mom and dad. For chromosome 2, you have a chromosome from dad and from mom. But now your DNA is duplicated, so you have two sides to that chromosome. So let's talk about what diploid, which we call 2N or two sets of chromosomes, versus haploid or one set of chromosomes, what those terms mean and what that looks like. So in human beings, nearly all cells have paired sets of chromosomes. You have one from dad and one from mom, again, for each of the 23 pairs. Pair 1, pair 2, I'm not showing you all 23 pairs here because that would take up a lot of space, but again, this cell with a chromosome from dad and from mom would be called diploid. Now, whether the DNA is unduplicated, as in here, or duplicated here, these are both of these examples, this and this, are both diploid because you have a chromosome from dad and from mom. Now here, if we just see a copy of the chromosomes from dad, which, for example, would be in a sperm cell, and you only have a copy of chromosome from dad, for example, we call that haploid. And that we call that 1N because there's just one set of each chromosome. There's one of chromosome 1, one of chromosome 2, and one of every other chromosome. So it's easy to get confused and see this double structure and think that that might be diploid. That's not the case. Whether you have two sister chromatids together or not does not make diploid or haploid. It's whether you have a copy from dad and mom is diploid. Dad only is haploid. So this is haploid. This is also haploid because there's only one set of each copy from dad in this example. So again, remember, it's not whether the chromosome is duplicated. It's not if you're here or here or here or here that makes haploid or diploid, but whether you have that copy from dad and from mom. Dad and mom copy is diploid. Dad copy only or mom copy only would be haploid. Okay, now that we've got some of those terms hopefully under our belt and a little bit more clear on what those mean, let's review what the goal of mitosis was and then review what the goal of this process we'll talk about this week, which is meiosis, review what that is. So we know what your cell is trying to accomplish as it goes through each of these types of cell division. So mitosis is when a diploid cell, so a normal cell in your body with a chromosome from your mom and from your dad, divides and produces two more diploid cells that are both identical to the original cell. So if you think of it, if your skin cell needs to replicate and your body's trying to grow more skin cells, you want every skin cell that gets produced to be just like the skin cell before it. So mitosis produces identical cells. Your cell goes through one cell division and produces two new cells that are identical to the original cell. So you go from 2N, set of chromosomes from mom and dad in the beginning cell, and then each new cell also is 2N, set of chromosomes from mom and dad. And again, that is mitosis. Now, meiosis is the way that your body produces sperm and egg cells or reproductive cells. And in this case, you start out with a diploid cell, so also 2N. You have a copy of each chromosome from mom and dad, but you're going to go through two different cell divisions, and you're going to end up with 4 new cells that are all 1N. So the sperm and egg cells all have one copy of each of those chromosomes. So mitosis starts with 2N, ends up with 2N. Meiosis starts with 2N and ends up with 1N, or starts with diploid and ends up haploid. Now the haploid cells that are produced through meiosis, another term for these is gametes. And gametes are just the reproductive cells. So in humans and a lot of other animals, this would be sperm and eggs. So female gametes are called eggs, male gametes are called sperm, and they're the reproductive cells of human beings and many other organisms. Remember, gametes, or sperm and eggs, are haploid. They have only one copy of each chromosome. Here's a haploid sperm, here's a haploid egg, and so we call them N or 1N because they're haploid and they have one set of each chromosome. And again, these cells are made through the process of meiosis. Now each eggs and sperm have one copy of each of the chromosome. Again, that's why they are haploid or N. And when the sperm and egg come together and the sperm dumps its DNA inside the egg and the egg gets fertilized, we call this new fertilized egg a zygote. And it has a copy now of the chromosomes from the sperm and from the egg. So one copy from the sperm, one copy from the egg. That fertilized zygote, or that fertilized egg now, is now 2N or diploid. So the reason that we need our sperm and eggs to be haploid, or to have one copy of each chromosome, is so that when the sperm and egg come together, that first cell produced in the new baby or the new offspring is 2N, and then that cell will divide over and over again through mitosis and stay 2N and grow an entire living organism that has each cell that is also 2N. So again, gametes are 1N or haploid, and the fertilized cell is 2N or diploid, and we call that fertilized cell a zygote. Let's look now, section 10.2, at the steps of meiosis and go over each of these steps in detail of how that cell division occurs to create these gametes. Okay, so this looks complicated in the picture, but let's just quickly go over what is going on here, and then we'll go over these steps in general. So in meiosis, we have two different cell divisions. We start out with our first cell. It goes through this whole process similar in steps to mitosis, a little bit different. We'll go over how that is, but you start out with one cell. It goes through a cell division. You end up with two cells. Those two cells each go through a new cell division, and you end up with 4 cells in the end. Now, if you remember, mitosis went through one division, you ended up with 2 cells in the end. Meiosis goes through meiosis 1 and meiosis 2, and you're going to end up in the end with 4 cells, either 4 egg cells or 4 sperm cells. And now we will go through each of these steps down here and go over what's happening in those steps. So before we can actually talk about meiosis, we have to talk about the interphase, which happens before meiosis, where the chromosomes duplicate. So to begin with, each cell has 2 sets of chromosomes, again 2N, or 46 chromosomes total, 23 from mom, 23 that you got from dad. So there's 46 chromosomes and 46 chromatids. This is what it looks like here. So we don't have the sister chromatids yet. We just have a copy of a chromosome from dad and a copy from mom. And during interphase, so before meiosis starts, the DNA is copied, and so then we end up with the sister chromatid layout here. So we have 2 sister chromatids making up each chromosome. So DNA here that's unduplicated gets copied, and now you have your replicated chromosomes. And so in every cell now that's getting ready to go through meiosis, you have 46 chromosomes still. So still 23 from mom and 23 from dad, but you now have 92 chromatids. So 92 is just double 46. So you have, if you were to count up all 46 chromosomes, 2 chromatids on each chromosome makes a total of 92 chromatids. So in interphase, the chromosomes duplicate, and on each slide I'll go through how many, whether we're at 2N or 1N, haploid or diploid, how many chromosomes we have and how many chromatids we have. So again, before interphase, the cell is is diploid, it's 2N. There are 46 chromosomes, 46 chromatids. The DNA duplicates. We are still at 2N. We still have 46 chromosomes, one of each type from mom and one of each type from dad. But now that the DNA is duplicated, we have 92 chromatids. So 2 chromatids on every single chromosome. Now we move into meiosis 1. So this is the first replication, and the first stage of that is called prophase. This is prophase right here. Remember, we have sister chromatids because our DNA is duplicated, and our chromosomes during prophase pair up. So the chromosome from mom and the chromosome from dad get next to each other for chromosome 1, chromosome from mom and dad for chromosome 2 get together, and all the 23 pairs, they arrange themselves so that the like chromosomes, or what they call homologous chromosomes, are paired up. And they do something special when they pair up here, and that they will actually cross over bits of the chromosome. Those legs of the chromosomes will sort of wrap around each other, and they will mix up their DNA. So segments of this chromosome will move on to this one, and segments from this chromosome will move on to this chromosome. They call this process crossing over, and this occurs during prophase. They also sometimes call it recombination, because the chromosomes and that DNA are getting recombined. So again, here's an overview of when you look at crossing over, what's happening. So the homologous chromosomes, or let's say chromosome 1 from dad and chromosome 1 from mom, they get close to each other. They sort of wrap the arms of the chromosome here, wrap over each other, and they exchange bits of DNA. So if you look at this chromatid right here, they call that a recombinant chromatid, and that's because it's been recombined. It has bits of DNA from the mom and the dad chromosome. This one over here did not get mixed up with the other chromosome. It has DNA still just from dad, so it's considered a non-recombinant chromatid. Now, this is an important step because it's what makes when you make the sperm and the eggs in the end, it's what makes them have a little bit of DNA. So if you're a girl and your body's making eggs, it's what makes your egg have a little bit of DNA from your mom and from your dad. If you're a boy and you're making sperm, it's what makes each of your sperm have a little bit of DNA from your mom and your dad. But since these chromosomes can wrap around each other and rearrange in any different combination, it's also what makes every egg or sperm you produce different. So if this process didn't occur, every egg and sperm that you made would just have a chromosome from one side of the chromosome from your mom, one side from your dad, and they would all be exactly the same. But this allows every sperm and egg you make to be a little bit different, and that's why every child that you have will be different. That's why you and your siblings are not exactly the same, because of this process right here. So this is a really important part of meiosis. Let's move on. We're still in meiosis 1 in that first division, and we're now at metaphase. Now metaphase is the same as it was in mitosis. The chromosomes line up here at the center of what they call the metaphase plate, and there's one chromosome on one side of the plate, one set of each chromosome, and one set of each chromosome on the other side of the plate. We still are diploid here since the chromosomes haven't divided yet, and we still have 46 chromosomes and 92 chromatids. Moving on to anaphase, the homologous pairs are then separated, so one of chromosome 1 goes to this side, one of chromosome 1 goes to this side, one of chromosome 2 goes to this side, one of chromosome 2 goes to this side, etc. for all 23 different pairs. And the sister chromatids remain attached. They do not separate from each other during this part of meiosis. So that's a little bit different than mitosis, which looked more like this here. So sister chromatids remain attached and separate to opposite sides of the cell during anaphase. Again, are still diploids, still 46 chromosomes, and still 92 chromatids. Now during telophase, this is when the cell membrane starts to pinch in, just like it did in mitosis. The cell is starting to divide, so the chromosomes are at the poles and the cytoplasm is dividing during telophase. Cytokinesis, again, is the splitting of that cell all the way through the middle and splitting into two different cells. Now so we've had diploid 46 chromosomes, 92 chromatids, but after that division, each of the new cells that forms after that division will be haploid. So it will have one copy of each chromosome. It will have a total of 23 chromosomes and 46 chromatids. So we've gone from this and we've split all those numbers exactly in half for our new cell, which makes sense because we literally took the cell and split it in half. Now let's look at meiosis 2. This is the second division of meiosis. And if you remember, there was a duplication of the chromosomes before meiosis 1. So in interphase, the chromosomes duplicated. This does not happen prior to meiosis 2. There's no duplication of DNA. First is prophase. This is very short. This is where sort of the parts get formed to start getting ready to pull the chromosomes apart. They call these, there's these things that they don't show on here, but these centrioles, little things that start making these fibers here, these purple fibers to help the chromatids pull apart and the chromosomes start getting arranged. And remember, at the beginning of meiosis, we have what we were left over with from meiosis 1. So we're haploid, one end, so that means we have 23 chromosomes, one copy of each chromosome, and 46 chromatids. Now in metaphase, just like in every other metaphase that we've seen, the chromosomes start to line up at the center plate. Still, nothing's changed here yet. In anaphase 2, the sister chromatids pull apart. So again, half of this chromosome 1 will move over here, half of chromosome 1 will move over here. Half of chromosome 2 moves to one side, half of chromosome 2 moves to the other side. And each of these separated sister chromatids becomes a full-fledged chromosome in this process. So instead of being 1 in 23 and 46 like we were before, we are now at 46 chromosomes again. So we were in the last step at 23 chromosomes, but now each of these halves of the chromosome acts as a single chromosome. So during anaphase 2, we are at haploid, 46 chromosomes, 46 chromatids. So during our next phase, telophase 2, the cytoplasm begins to pinch in here. And finally, it pinches in all the way through, and we have cytokinesis, and 4 cells have now been produced. Each of those cells is haploid, as it has been all through meiosis 2. And each of these cells now, in the prior cell, we had 46 chromosomes and 46 chromatids. Now we've split that in half, and we have 23 chromosomes and 23 chromatids in each new haploid cell that we've made. And again, these would be each of these would either be an egg sperm, or each of these would be an egg cell, or each of these would be a sperm cell. So if you want to come back and look at some point, you don't need to memorize this, but if you were confused on how many chromosomes and chromatids you have during each phase, this is a good table to come back to and just review that. It does get complicated where things duplicate and divide, and so this is just a good overview of our chromosome count of each phase in meiosis and our chromatid count in each phase of meiosis. Now, the main significance of meiosis is that meiosis generates diversity by ensuring that the gametes it gives rise to will differ genetically from one another. And what this means is, like we talked about earlier, that every egg cell or sperm cell that you produce is different from every other egg or sperm cell you produce due to that crossing over. And so you've essentially, through meiosis, you're mixing up your DNA and you're producing all these different types of sperm or egg cells. And this helps ensure that all the different genes you have get passed on to your offspring randomly. So not only by making sperm or egg cells that are all different from each other are you making these diverse sperm and egg cells, but when organisms mix together a sperm and egg, you are mixing the DNA from 2 completely different organisms together and mixing their DNA up so that when an offspring is created, their offspring is different from either one of them. So every offspring that gets created through sexual reproduction is different. Now, this may seem like, how could you do this any other way? But in some organisms, such as bacteria, the cells just divide and make new organisms that way. And so every offspring of that bacteria that divides is just a clone of itself. And so there is no diversity that comes through that process. We call that asexual reproduction. But here in meiosis, we're dealing with sexual reproduction, and we are dealing with mixing DNA from 2 different organisms, male and female, together to produce a genetically different offspring. Next, let's look at how meiosis determines the sex outcome of offspring. And the way that this happens is through the sperm cells. And the sperm cells, a regular adult male body cell has an X chromosome and a Y chromosome. And so the male can pass on either an X chromosome, which will make a female producing sperm, or a Y chromosome, which will make a male producing sperm. We'll go over to that on the next couple slides. So we've looked at this before, but here's a look of what all the chromosomes of a male and a female look like. And males and females have 22 pairs of what they call autosomes, which are all your other chromosomes except the X and Y chromosome. So males and females both have very similar chromosomes for all the other chromosomes, and the main difference is when you look at these other 2 chromosomes, which we call the sex chromosomes, and those are the X and Y chromosomes. So chromosomes 1 through 22 on both the male and female we call autosomes, and then the X and Y chromosomes we call sex chromosomes or sex determining chromosomes. Now, a female starts out with their chromosomes looking like this, 22 autosomes, pairs of their other chromosomes, and then one pair of X chromosomes. So a female has two X chromosomes. So when a female cell starts to undergo meiosis, it has two X chromosomes in it, goes through meiosis 1, each of those X chromosomes splits into a different cell, goes through another division, and there's an X chromosome in each of those new cells. So after meiosis, the female has gone from XX to producing two X chromosomes to producing 4 egg cells, all with an X chromosome in them. So human males have 22 autosomes. They have one X chromosome and one Y chromosome. So when a human male cell goes, or not even a human male, but a male from any organism goes under meiosis, the male, all of these, remember, on each of these pairs, one chromosome will go into one of the new cells, one chromosome will go into the other new cell. Now in the X and Y, that's the same thing. So the X chromosome will go into one new sperm cell, and the Y chromosome will go into another new sperm cell. So when a male undergoes meiosis, he either makes sperm with an X chromosome or sperm with a Y chromosome. Let's just look at this again. Here's our steps of meiosis. We have our father starting with an X and a Y chromosome in his normal body cells. When he undergoes the first round of meiosis, the X and the Y split apart into two separate chromosomes. And in meiosis 2, he has another separation of cells with an X chromosome going into each new cell from this one, and a Y chromosome going into each new cell from this cell. And so at the end of meiosis, the male has produced 2 sperm cells with X's in them and 2 sperm cells with Y's in them. Now when a sperm and an egg come together, the mother will always give an X chromosome into her egg. If the father gives an X chromosome, and if so a sperm with an X chromosome fertilizes that egg, then you will get a daughter or a female. So if the father's chromosome that fertilizes the egg is an X chromosome, you get a female. The mother, even if she's making a son, still gives an X chromosome. It's the father's X or Y chromosome that determines whether you get male or female. So in the case of a male, a sperm with a Y in it is the sperm that fertilizes the egg, and you end up with an X and a Y chromosome in that new cell, which will make a son or a male. So a female, once again, will always give an X chromosome, and it's the male's X or Y chromosome that determines whether the offspring is a male or a female. Next is section 10.5, gamete formation in humans. Not all of your body cells can undergo meiosis to produce sperm or egg cells. The specialized cells that undergo meiosis are oogonia for females, and oogonia are the type of cells that undergo meiosis to become eggs, and spermatogonia are the special type of cells that undergo meiosis to become sperm. Unlike a male, female gamete formation begins with the oogonia, most or all of which are produced prior to the birth of the female. So the eggs that the female is born with are all formed before she's even born. So the eggs that she has are what she'll have for life, and the male, on the other hand, will continue to produce sperm throughout their life and continuously be making new sperm, whereas again the female is born with all of the eggs that she has or will have in her lifetime. Now, all male cells that undergo meiosis, as you can see here, here's a male cell undergoing meiosis and undergoing the first division and the second division, and all of those cells become sperm. Now in a female, the female egg or oocyte undergoes the first division here, and one of these new cells out of that division will get most of the cytoplasm. That cell that gets most of the cytoplasm will again divide, and only one of those cells will actually become an egg. The other cell that didn't get much cytoplasm will divide, and this other cell here that also didn't get a lot of cytoplasm, they will end up as what they call polar bodies, and they will just break down and not become an egg. So when a female oocyte undergoes meiosis, only one of those cells, only one of the 4 cells out of meiosis will actually become an egg that is possible, has the possibility of being fertilized. All right, so all of these pre-egg cells, or these oocytes, sit in the female ovaries ever since the female is born in prophase 1 of meiosis. Now, the oocytes start getting released from the ovaries after puberty, and typically one oocyte will get released per month, and only that oocyte that gets released will finish meiosis 1 during that month. All of the other oocytes sit in the ovaries and just wait there and don't complete meiosis 1 until they are also eventually released from the ovary. Now, the oocyte will only complete meiosis 2 if it's fertilized by a sperm cell. Next up, we have section 10.6 called Life Cycles, Humans and Other Organisms, and we'll look at how some other organisms reproduce. Now, not all reproduction, as we mentioned, is sexual reproduction, meaning reproduction that works through fusion of 2 reproductive cells, such as an egg and a sperm. Other organisms can use other types of reproduction that don't involve an egg and a sperm. Most types of organisms are capable of asexual reproduction. This is rare among more complex organisms, and it's never carried out by mammals or birds. And asexual reproduction would mean reproduction that does not involve, again, the joining of gametes such as egg and sperm. Some examples of asexual reproduction, which we'll talk about next, are binary fission, vegetative reproduction, and regeneration. Bacteria reproduce through one form of asexual reproduction called binary fission. Now, bacteria only has a single chromosome, so in binary fission it replicates a single chromosome and then divides into 2, and this process is called binary fission. It's how bacteria asexually reproduce. So every offspring bacteria is a clone of the parent bacteria. Plants can do something called vegetative reproduction, where a part of the plant grows off and produces, say, a new tree. So like a new bud off of a root can sprout and produce a new plant. Other organisms such as worms and sea stars can carry out regeneration, in which a new complete organism can be formed from a portion of an existing one. So let's say a piece of a starfish or a sea star breaking off and becoming a new sea star. That would be an example, or a worm splitting into 2 and each part of the worm becoming a new worm. These would be examples of regeneration. And here's a picture of a sea star. So it had one piece of the sea star here from the original organism that broke off, and it is regrowing a new sea star. And we've reached the end of our Chapter 10 lecture. Please let me know if you have any questions as you go about your week, and I will be in touch with you again next week for Chapter 11.